Hi, hello to all the NEET aspirants. Hope you are very much busy with your preparation. So, I got you the best shortcut methods and memory hacks, and now we are going to deal with the NEET Botany 2023 paper with all the shortcuts possible and the memory hacks. Okay. So, yes, if you look into the first question, which is of assertion reason type, what we have to do when we are getting assertion reason type of questions. Clearly see beta, the question is from plant kingdom and the first stage of gametophyte in the life cycle of moss is protonema stage. Do remember, don't get confused. Most of the students get confused with moss and fern. Okay. Moss, you have to immediately remember it is what? A bryophyte. Okay. It is a bryophyte. Fern is a pteridophyte. Okay. It is a pteridophyte. And what is the primary gametophytic structure in a moss? The primary gametophytic structure is protonema. Okay, the primary gametophytic structure is protonema. The primary gametophytic structure of fern is what? Prothallus. It is what, Nana? Prothallus. Clear? Remember this. And here the first stage of gametophyte in the life cycle of moss is protonema, correct? And protonema develops directly from spores produced in the capsule. If you recall the moss diagram in our plant kingdom, wherein from the leafy gametophytic structure, foot seta capsule, that is the sporophyte that is emerged, right? This is capsule, this is foot, and this is seta. Okay, this leafy gametophyte, rhizoids, all these are haploid gametophytic structures. Seta, foot, capsule, these are diploid sporophytes, right? Understood, Nana? So, inside capsule what happens? Spore mother cells which are diploid, they undergo reductional division and they form haploid spores. They undergo reductional division and they form haploid spores inside the capsule itself. And once the spores are formed, they fall onto the substrate and that gives rise to a new moss. So, here what is the correct answer, Nana? Both A and R are correct and R is correct explanation for A. Option 1 is the correct answer. Next question, if you look into, in angiosperms, okay, in angiosperms, the haploid, diploid and in angiosperms, the haploid, diploid and triploid structures of fertilized embryo sac. He is asking you of a fertilized embryo sac. Do remember, after fertilization in an embryo sac has two ends, right? Micropylar end and chelacal end. At chelacal end you have three antipodals and at the center you have polar nuclei, or you will call it as a central cell. Towards the micropyle you have synergids with filiform apparatus and an egg cell. Okay, synergids with filiform apparatus and an egg cell. Okay, just a second. Yes. So, after fertilization, one male gamete will come and fuse with the egg cell forming diploid zygote. One male gamete will come and fuse with the polar nuclei forming triploid endosperm, and this is a diploid zygote. Okay, and antipodals are haploid. First, before fertilization, embryo sac is a haploid female gametophytic structure in angiosperm with 7-celled 8-nucleated condition. In the fertilization or after fertilization in a fertilized embryo, you can see diploid zygote, haploid synergids and antipodals and triploid endosperm. So, what will be our answer here, Nana? Haploid is haploid synergids, diploid zygote and triploid primary endosperm nucleus, that is third option. Next, which hormone promotes internode or petiole elongation in deep rice water? In deep rice water, internode or petiole elongation is done by which phytohormone, Nana? Ethylene. Okay. If it is in the sugar cane, or maize or mesophytic plants, then it is gibberellic acid that increases the internodal elongation in the stem. Okay, kinetin is the synthetic cytokinin, okay, artificial or synthetic cytokinin. 2,4-D, this is the synthetic auxin which acts as the best weedicide, and this will kill mostly dicotyledonous weeds. Okay. Next, large colorful fragrant flowers with nectar are seen in insect pollinated plants. They are seen in what, Nana? Insect pollinated plants. This is from pollination of sexual reproduction in flowering plants. Then the phenomenon of pleiotropism refers to, what? Pleiotropism, multiple allelism, incomplete dominance, codominance, these are the exceptions for Mendelian monohybrid cross. We all know that, right? So the best example for pleiotropy is what, Nana? One gene is showing multiple phenotypic expressions, and examples include phenylketonuria, starch synthesis. These are few examples for pleiotropism, and here in pleiotropism single gene affects multiple phenotypic expressions. Next, if you see, upon exposure of UV radiation, this in the successive years we can see the question from the DNA staining. So here under UV radiation, DNA stained with ethidium bromide will show what color? It will show bright orange color. Okay, we have dealt it so many times. And then axial placentation. Of course, we all know we will get definitely the questions from either one, definitely two questions to pakka from morphology of flowering plants with respect to the examples. All the examples of vegetative, especially for floral characters, placentation, aestivation, definitely from examples you will get the questions beta. In our channel, in shorts you can clearly see the memory hacks also to remember all the examples. Just go through it once, okay. So axial placentation is observed in China rose, petunia and lemon. Option 4 is the correct answer. Next, the process of appearance of recombination nodules occurs at which substage in prophase 1? We all know prophase has 5 substages: leptotene, zygotene, pachytene, diplotene and diakinesis, right? So recombination nodules forms in which phase, Nana? Pachytene stage, that is third substage of prophase 1. Next, reaction center of PS2 in the thylakoid lamellae or thylakoid membrane has an absorption maxima at P680 or 680 nanometers. If it is photosystem 1, then the absorption maxima is 700 nanometers. Clear, Nana? Next, unequivocal proof that DNA is the genetic material was first said by Hershey and Chase. Unequivocal proof is given by Hershey and Chase experiment. This also I am seeing second time in the PYQs, okay. That is by the bacteriophage experiment, okay. So that is given by Hershey and Chase. Next, among eukaryotes, replication of DNA inside nucleus and centriole duplication in the cytoplasm takes place in the S phase. In which phase, Nana? S phase. Next, in tissue culture experiments, leaf mesophyll cells are put in the culture medium to form the callus. This phenomena may be called as, okay. So in tissue culture what is happening? Leaf mesophyll is a differentiated tissue. This is a differentiated tissue, right? And this leaf, when it is kept in a tissue culture, this is again gaining the activity of cell division. This is again gaining the activity of cell division, and it starts dividing and then it forms a callus. When you give equal proportion of auxin and cytokinin, callus formation takes place, okay. So here again, as they are gaining back their division activity, you will call it as meristematic activity, right. So this is losing the capacity of differentiation, again getting meristematic. So this is de-differentiation. What is it, Nana? This is de-differentiation, okay. Next, cellulose doesn't form blue color with iodine. It is not forming blue color with iodine iodine? This is because cellulose is a basic homopolymer of glucose which is formed by 1,4 glycosidic bonds. Okay. So, yes, here as it does not have complex helices and hence cannot hold iodine bonds. That is why it will not show blue color when it is added with iodine, like how starch is showing. Okay. Next. So, here the option is 3. Then if you see, spraying of which of the following phytohormone on juvenile conifers helps in harvesting the maturity period that leads to seed production? Always early seed production, or for juvenile conifers means you have to remember that is gibberellic acid. What is it, Nana? Gibberellic acid. And which of the following stages of meiosis involves division of centromere? Centromere division takes place, or centromere splitting takes place in anaphase of mitosis and anaphase 2 of meiosis. Anaphase 2 of meiosis. Clear? And next, family Fabaceae differs from Solanaceae and Liliaceae. Okay. With respect to the stamens, pick out the characteristics specific to the family Fabaceae. Pick out the characteristics specific to the family Fabaceae, but not found in Solanaceae or Liliaceae. Okay, which is only seen in Fabaceae, that we have to look into. That is Diadelphus and Dithecus anther. Diadelphus anther you can see only in the Fabaceae, that you cannot see in Solanaceae or Liliaceae. In Solanaceae and Liliaceae, what you will see? Adhesion of andrecium, wherein andrecium is attached to the corolla. Okay, that is epipetalous condition. Andrecium attached to the perianth, that is epiphyllous condition in Liliaceae. Clear, Nana? So, here what is the option? Answer 1. Expressed sequence tags. I can find the question on sequence annotation and expressed sequence tags in the successive years continuously. So, look into it keenly. Expressed sequence tags refers to that all genes that are expressed as RNA, all the expressed sequences or all the coding sequences comes under expressed sequence tags. Next, how many ATP and NADPH2 are required for the synthesis of one glucose molecule in Kelvin cycle? Of course, we all know. Let us quickly revise the Kelvin cycle once. 6 CO2, sorry, for one glucose formation, okay, in Kelvin cycle, for one glucose formation, we need 6 Kelvin cycles, that is 6 carbon dioxides have to be fixed. Yes or no? So, it requires 6 carbon dioxides, 6 ATP and 6 6 carbon dioxides means it requires, for one carbon dioxide, let us calculate first. For one carbon dioxide fixation, 2 ATP and 2 NADPH, 2 ATP and 2 NADPH are utilized during reduction, and one more ATP at the time of regeneration. But for the formation of one glucose, we require 6 carbon dioxides, 6 into 3, that is 18 ATP, and 6 into 2, that is 12 NADPH. These are required. So, here what will be our answer, Nana? 18 ATP and 12 NADPH. Option 2 is the correct answer. Next, in the equation, GPP minus R is equal to net primary productivity. What is R over here? R is respiratory loss. From this formula also we are getting the question, this is second time. Okay. Now, what is the role of RNA polymerase 3? From either RNA polymerase 1, 2, 3, definitely I can see the question in the successive years beta. Please once show keen interest on that, wherein RNA polymerase 3 will synthesize what? tRNA, 5SR RNA and SN RNA. So, that is option 2. If you look into rRNA, that is synthesized by RNA polymerase 1. Okay, mRNA is synthesized by RNA polymerase 2. Okay. Next, what is the function of tassels on the corn cob? Tassels are present. Tassels are present on the female plant. Okay, tassels are present on the female plant. This is able to attract the pollen grains. This is able to what? Attract the pollen grains, trap the pollen grains. Okay, tassels in corn cob is to trap the pollen grains. Next, identify the pair of heterosporous pteridophyte. For this, the best mnemonic I can give you is Mars. Okay, for heterosporous condition, which is heterosporous condition first observed in pteridophytes only, right? So, best example is Mars, Marsilia, Azolla, Selaginella and Salvinia. Okay, Selaginella and Salvinia, option 2. Okay. Next, endarch and exarch xylem, this is from anatomy. Endarch and exarch are the terms often used for describing the position of secondary xylem. Wrong. Endarch and exarch are used to describe the position of primary xylem. It is used to describe the position of what, Nana? Primary xylem. And if you look at, so this is wrong. Next, if you look into statement 2, exarch condition is the most common feature in the root system. Endarch is most common in the shoot system. So here what is the correct answer? Statement 1 is wrong and 2 is true. That is option 4. Okay. And then frequency of recombination between the gene pairs on the same chromosome as a measure of distance between the genes to the map their position on the chromosome, to map their position on the chromosome, was used for the first time by Alfred Stuyvesant. Okay, it is used for the first time by Alfred Stuyvesant. Next, how many different proteins does ribosome consist of? They consist of 80 different proteins. Okay, they consist of how many, Nana? 80 different proteins. And then if you look into again assertion reason question, that is from plant physiology, wherein ATP is utilized at 2 steps in glycolysis, respiration and plants. Okay, yes, they are utilized in 2 steps, that is at first step and third step, no beta. So first ATP used in converting glucose to glucose 6-phosphate and second conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. This is a rate limiting step. Third step is what? Rate limiting step. So here both A and R are true and R is the correct explanation of A. So that is option 1. As this is explaining the assertion, we have to go for correct explanation of A. Most of the students will get doubt with the assertion reason question only, that too when both of them are correct. You have to add some prepositions after assertion like is, have, but, okay, like that. If you feel like reason is explaining assertion in that way, once you read reason twice after assertion, if you feel like reason is correctly explaining the assertion, then only you go for the option 1. If you look here, he said ATP is used at 2 steps in glycolysis. At which steps? At which steps means at first step and at the third step. As this is explaining, we have to go for the option 1. Then if we see, which of the following statements is about Klinefelter syndrome? Okay, which is about Klinefelter syndrome. So Klinefelter syndrome, if you see, Langdon Down, this is, he discovered that for Down syndrome, and individual has overall masculine development. Klinefelter syndrome individual has masculine development. However, feminine development is also expressed in Klinefelter syndrome. Next, the affected individuals have short stature, that is in Turner syndrome, and physical, psychomotor and mental development, that is seen in the Down syndrome. Next, such individuals, Klinefelter syndrome or Turner syndrome, both the individuals are sterile. Klinefelter syndrome are seen in whom, beta? Males. Turner syndrome is seen in females. So here both, B and E both are correct. So this is option 3. Okay. Next, this is again match the following question from this chapter, plant physiology, respiration in plants, where oxidative decarboxylation is having an enzyme. This is mediated by the enzyme, and this takes place in mitochondrial matrix immediately after glycolysis in aerobic respiration, wherein oxidative decarboxylation needs the enzyme pyruvate dehydrogenase. Glycolysis is also called as EMP pathway after the scientists Emden, Meyerhoff and Parnas. Next, oxidative phosphorylation. Oxidative phosphorylation is electron transport system, Nana. Photophosphorylation that you will see in photosynthesis. Photophosphorylation that you will see in photosynthesis. Here oxidative phosphorylation, that is ETS, electron transport system, that takes place in inner mitochondrial membrane. Tricarboxylic acid cycle, that is TCA cycle, the very first enzyme in that is citrate synthase that will synthesize citric acid. Okay. So, here what is our answer? A2, B4, C3 and D1, option 4 is the correct answer. Clear, Nana? Next, if you see, assertion. A flower is defined as a modified shoot within shoot apical meristem changes to floral meristem. Shoot apical meristem will change to floral meristem. How is it changing? Again look into the reason properly. How is it changing? It is changing like internode of the shoot will get condensed to produce different floral appendages laterally at successive nodes instead of leaves. Okay, that is how a shoot apical meristem is modified to floral meristem and then gives rise to floral primordia. So, here A and R are true and R is the correct explanation of A. Next, assertion. In gymnosperms, the pollen grains are released from the microsporangium and carried by air currents. True. Air currents carry the pollen grain to the mouth of archegonia, where male gametes are discharged and pollen tube is not formed. No, pollen tube is formed and this is siphonogamous oogame. Siphonogamous oogame. Okay. In Rhyo and Pterido, if you see, that is zooidogamous oogame, but here it is siphonogamous oogame. Once the pollen grains are discharged at the mouth of the archegonia, pollen tube development takes place. So, here again A is correct and reason is false. Okay. A is true and reason is false. Option 3 is correct, Nana. Next, which of the following is combination required for chemiosmosis? For chemiosmosis, we need proton pump, proton gradient, membrane and ATP synthase. Membrane, proton pump, proton gradient and ATP synthase, that is F0, F1 pump. Okay. So, here it is 1, option 1. Next, malonate inhibits growth of pathogenic bacteria by inhibiting the activity of. This also I can see second time or third time. Malonate will inhibit the activity of succinate dehydrogenase, that is option 1. Next, M phase. From cell cycle, so definitely you will get either 2 or 3 questions in the NEET. Okay. So, here if you see, Nana, M phase. Okay. M phase is the division phase or equational phase, you can call it as, that is 4. G2 phase, protein synthesis will happen. Quiescent stage is the inactive stage. G1 phase, if you see, in G1 phase, interval between mitosis and meiosis, that is option 3. So, here if you clearly see, once taking down the cell cycle, this is G1 phase, S phase, G2 phase and this is M phase. Okay. So, here G1, S, G2 and M phase. G1 phase is in between M phase and S phase. G1 phase is in between M phase and S phase, where G1 phase is the interval between mitosis and initiation of DNA synthesis. So, here what is the option, Nana? A is 4, B is 2, C is 2, B is 1, C is 2 and D is 3. Option 3 is the correct answer. Next, which of the following symbols represent mating between relatives? Okay, that is 2 lines in between male and female. that you have to see. Okay, if this is in the pedigree chart, then it is with 2 lines between male and female. If it is normal mating, then this will be your answer. 2 normal parents affected offspring, then this is your answer. This is sex unspecified. Okay, male, female, sex unspecified, all of them are deceased, then your answer will be option 4. Next question, if you see, which of the following is not associated as a part of endomembrane system? Endomembrane system includes all the single celled organelles, which function in a coordinated manner, like what are the endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. So these 4 will be there. Okay, what are they, Nana? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. These are the 4 organelles which are included in the endomembrane system. So here what will be your answer? Endoplasmic reticulum comes under endomembrane system, Golgi bodies also, not mitochondria, chloroplast or peroxisomes. So here the answer will be A, C and E, that is option 2. Next, a protein is imagined as a line, the left end represents first amino acid and right the second amino acid, true, C terminal and N terminal. Adult human hemoglobin has 4 subunits, 2 alpha and 2 beta subunits, that also is correct. Okay, so both statement 1 and statement 2 are true. Okay, then gene A, Y and IZ, if you see, gene A codes for what? The enzyme beta galactosidase. A is 1, Z is coding for, okay, gene Z is coding for beta galactosidase, Y for permease, A for trans-acetylase and I repressor gene. Once quickly take down the structure of Lacoperon model from 5 prime to 3 prime end, where you have promoter I gene followed by promoter, 3 structural genes and lastly terminator. If you clearly look into first, I gene will release out repressor. To promoter region, RNA polymerase will bind Z, okay, Z, Y, A genes. Z is coding for beta galactosidase, Y for permease and A for trans-acetylase. So here what will be your answer, Nana? A is 2, B is 3, C is 4 and D is 1. Option 2 is the correct answer. Next, broad palm and single palm crevice is visible in a person suffering from Down syndrome. Okay, that is what? Down syndrome. Next, again this is from cell cycle and cell division chapter. Tetrad formation is seen during leptotene. Wrong. Tetrad or bivalent or homologous chromosomes or synaptonemal complex formation, all this you can see in zygotene. Okay. During anaphase, the centromeres split and chromatids will separate. Yes, true. Next, terminalization takes place during packaging. Wrong. Terminalization of synaptonemal complex takes place in diplotin. Terminalization of chiasmata takes place in diakinesis. Nucleolus, Golgi complex and ER are reformed during telophase. True. Crossing over takes place between sister chromatids of homologous chromosomes. True. Crossing over will take place between sister chromatids. Wrong. Crossing over always takes place between non-sister chromatids of homologous chromosomes. Okay. See, these two are sister chromatids, these two are sister chromatids, but crossover takes place between two non-sister chromatids of homologous chromosomes. So here what is the correct statements? Correct statements they are asking for. Correct statement is, okay, B and D, okay, option 2. Next, this if you look into, we have solved it in the MalBio PYQ session also. Which one of the following is sequence on corresponding coding strand? If the sequence on mRNA formed is as follows. If you clearly see, Nana, they have asked you for the coding strand, giving you mRNA strand. If you remember, mRNA strand will be complementary to template strand. If this is template and this is coding strand from 5 prime to 3 prime and this is from 5 to 3. Okay. mRNA strand synthesis will be forming from 5 prime to 3 prime. Okay. This will be complementary to the template strand, complementary to the template strand, and mRNA will be same as that of coding strand, except here if U is there, here T will be there. That's it. Here in the place of uracil, thymine will be there. Same you apply here. If you see AUC at 5 prime, okay, at 5 prime ATC, next GAT, CGAT, 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 CG, CGAT, CG, yes. So, what will be your correct answer? Option 3. Okay, this will be complementary to the coding strand has given, and mRNA will be same as that coding strand is asked. Okay, so mRNA will be same as that of the coding strand. Okay, and if they are asking you for complementary, complementary means same like this. If they are asking you for take down the template strand giving you the mRNA strand, then you have to code for a complementary sequence for this. Okay, like that you have to do it better. But in this question, what did they ask you? They have given you the mRNA strand and asking us to find out the coding strand. Okay, except in the U place, thymine will be there. That's it. So, with this we have completed 2023 question paper. In the coming session we will do 2024 question paper, and then the best sessions possible for all the diagram based questions. Okay. So, stay tuned and all the very best for your exam.